And I want to thank the organizers for inviting me to this really extraordinarily interesting meeting, and in addition for giving me the opportunity to visit this wonderful country for the first time. So I want to thank all four organizers for both of those reasons. My laboratory does have diverse interests, but those interests by and large do not include stem cells or differentiation, which is interesting given that's the topic of this particular conference. Most of our interest centers around morphogenesis in one form or another, either morphogenesis of an organism during embryogenesis or changes in morphogenic programs to generate diversity that we find around us in nature. Today I'm going to talk about the morphogenesis of one particular structure, which are the villi of the intestine. Now you all know that these beautiful structures on the luminal side of the intestine form to increase the surface area of the gut, to give more surface area for absorption of nutrients as nutrients transit through the gut. And if one wants to think about how these arise in the first place, that is to say morphogenesis, there are actually two things that come to mind. And this is two panels from a larger set of panels in a review article that was published in Nature Reviews Genetics, showing the state of the early development of the gut in a mouse or a chicken or a human, and the mature gut at the birth or hatching in the case of a bird. And there are two things that have changed. Number one, you go from a smooth lumen to having these projections, these villi, these fingers reaching in. So how do you form such structures? How does that morphogenesis arise? Second thing is, to a first approximation, the early epithelium of the embryonic gut is entirely stem cells. All of these cells are proliferating, all of them are self-renewing. Yet, by birth, the stem cells are localized at the base of the villi, and subsequently post-birth they'll burrow down and form what are called the crypts, as you all know. So the second issue is, how do you get the stem cells specifically in the right location? And it turns out that not only are these linked, but they're in fact, one is dependent upon the other, as you'll see. So I want to start out by trying to understand the morphogenesis of this structure. So it's been known for a long time that the human villi, and this is not something that I would have guessed if I hadn't read the literature, they don't form by growing out from the floor of the lumen in a simple way, but rather initially, embryonically, you form ridges that then zigzag, and the zigzags segment to form ridges, to form villi. So the process has been described morphologically. Now, interestingly, and this is going to, I'll come back similar to what Alan talked about in the last talk, there's a seeming exception that we're going to have to address and show really isn't an exception, and that is that according to literature, that's not the way rodents do it. That is the way humans do it. Cattle, various other mammals form villi by forming zigzagged ridges that then segment. Birds do it the same way, as I'll show in the next slide. But rodents are the outliers. So if you want to have, this is taken from a paper in 1982, the mouse intestine does not start by forming previllus ridges that segment. So if you want to understand how this occurs in humans, the rodent's actually a lousy model. The chick, in contrast, is a very good model, and we do a lot of our work in the chick embryo. These are pictures from a 1958 publication. These are from our laboratory. But you can see there's a progression over time from a smooth lumen to the formation of ridges running the length of the entire intestine. Then these ridges zigzag in parallel, and then the ridges segment to give rise to villi. So we want to understand this process, and I should say that to begin with, these ridges form sequentially. So first you get one ridge, then you get 3, then you get 4, then you get 5 over time, until by 11 or 12 you have the full set of ridges that then subsequently go on to zigzag. Now, the formation of ridges in a luminal tube is something that's actually been very well studied, largely in the context of the trachea. And that's because the trachea, the process of forming ridges is very important for having an open airway. And in a normal trachea, you have a very thin epithelium. But what happens in the case of asthma is the epithelium gets thicker. And this turns out to be the cause of the problem with breathing in the case of asthma. And to understand that, you have to understand the process, which has been very well modeled. It's called epithelial buckling. And what it basically consists of is a continual growth of a thin layer on an elastic substrate that's not growing as quickly. And in asthma, the problem is not that the airways, that the tissue becomes so thick that the tissue itself swells and clogs things. What happens is by changing the thickness of the epithelial layer just a little bit, you dramatically change the pattern of buckling, and the folding then completely shuts off the airways. So it's been very well studied in that context. So Amy Shire, a graduate student who did almost all of the work I'm going to be talking about today, decided to investigate this and to see if that was what was going on in the intestine. We thought, well, we know what the two layers should be. The thin layer would be the endoderm, and then you've got all this mesenchyme. So she simply separated them, and when she did so, there was no change. So you take off at a ridge stage, you take off the epithelium, and the ridges remain. At a zigzag stage, you take off the epithelium, the zigzags remain. So that's not what's going on. So if it's not that the epithelium is a thin layer on an elastic substrate, what is happening? So that made us go back and think a little bit more about the structure of the intestine. Now, the mature intestine not only has villi, but underneath that there's a submucosal layer, sort of a mesenchymal layer, fibroblastic layer. And then you have, as you all know, several layers of muscles. Muscles that are circumferential around the outside, and muscles that are longitudinal running this way. It turns out the timing of those muscle formation has a very interesting pattern, where the time when the first row of circumferential muscles form is precisely when the ridges form. And you can see that here. Here you have a series of different stages during ridge formation, and during this time you have the differentiation of the smooth muscle, as shown by smooth muscle actin. Then, exactly at the time when that starts to zigzag, E15, embryonic day 15 in the chick, is when the next layer forms, which is the longitudinal muscles that run in and out of the plane of the blackboard here. And then exactly when those start to segment into villi is when you get the third layer of longitudinal muscles right underneath the epithelial layer. So we want to try and look at this. We're going to start by exploring this in the context of ridges. Now, to do the types of experiments that we want to investigate, or any experiment, if you want to do an experiment, you need to have the right people in your lab to do the work. In this case, we had the right person in Amy because she has very nice hair. Now, I don't know what nice hair means to stem cell people, since I've never really discussed it with any of them. But I can tell you what it means to an embryologist. It means two things. Number one, it's straight. Now, number two, it's oblong and flattened in shape. So many of the people here are Han Chinese. Han Chinese have very straight hair, but they have large round hair. No good for this. But if your hair is like this, you can turn this into a knife. You put the hair at the end of a pipette and then cut with the edge of it, and you can now just cut around, separating the tissues like butter. So with your hair-based knife, you can separate the muscle layer, which is on the outside, from the inner layer now of both the epithelium and the non-muscle mesenchyme. And if you do that, immediately things open up. Immediately, the outer layer stays the same size, but the inner layer unfolds. So all of this was in here. You lose any sorts of ridges, and instead you get two circles. So that suggests a model where the muscle, when it differentiates, forms a stiff barrier, so that as these tissues continue to proliferate, they're forced to buckle inward. So is that indeed the case? Well, to test that, we wanted to set up an in vitro system. So the first thing Amy did was show that in vitro she can culture guts, and after 48 hours, starting from a stage where there are no ridges, she can get the same sorts of ridges that you'd have at that same time if those guts had been left in vivo. So now she can repeat this in the presence of either of two drugs that block smooth muscle differentiation. And we're using two different drugs that block two different pathways to take care of off-target effects. If you get the same phenotype with both, it's probably because of their effect on smooth muscle. And then the prediction would be, if the smooth muscle is simply a barrier, then a barrier to expansion, then if you get rid of the smooth muscle, the tube should expand radially because it no longer has that barrier, and no ridges will form. And that's exactly what we see. So here's a fresh E8 gut with 3 ridges. Here, and smooth muscle, first smooth muscle layer. Take an E6 gut that has no smooth muscle, culturing it for 2 days, so it's at an age E8. You get the same 3 ridges with the smooth muscle. But if you block smooth muscle differentiation, you do not get the ridges, so you fail to get ridges. And now there are 2 reasons that could be happening, of course. Either because you've lost the barrier and the whole thing's expanding, or alternatively, you could argue that all you're doing is blocking proliferation of the inner tissues, and therefore you don't have the buckling. But there's no change in the proliferation of the tissues with either of these drugs, and indeed it does expand. So we think that this model is probably right, but if it is, then one can make a further prediction. If the smooth muscle is simply acting as a barrier, and in the presence of that barrier you don't get expansion, and if you don't get expansion, then you don't form ridges, then you should be able to restore the ridges by putting in some other type of barrier. And so here we are in China, which is the birthplace of silk. So it turns out, having tried lots of different types of barriers, the barrier that worked the best was silk. So we're using spun silk tubes from David Kaplan's group at Tufts. They're porous, so nutrients can get through. You can spin them at any diameter you want, and you can spin them as thick as you want, so therefore as stiff as you want. So David Kaplan's group made us tubes. If you make a tube that's larger than the diameter of the gut and put a gut in there, then there's no new barrier. Nutrients get through without any problem. The gut expands, and if you have the drugs that prevent smooth muscle differentiation, you get no ridges. But with the same drug, so you should get no ridges, if you now constrain expansion with a tube that fits tightly around it, you get the same sort of buckling that you would have had otherwise. So we think that the smooth muscle isn't contracting, it's not sending signals, it's simply a barrier. What about the zigzags? So zigzags, you have the ridges now, and now after this you don't get any more ridges, but all of a sudden they all become zigzags. Well, how you get zigzags in tubes and in nature is something that was actually explored by a physicist, Mahadevan's group at Harvard, in the context of leaves, actually. And what he found, using various physical models and mathematical modeling, is that the zigzag pattern, which is similar to a Japanese origami pattern, is obtained when you have a thin layer that's elastically supported, but now is compressed biaxially. Biaxial, two perpendicular directions. That's exactly what you would get with the longitudinal muscle and the circumferential muscle. So we then just looked and said, okay, are these two, are the circumferential and longitudinal muscles necessary to get the zigzags? So we repeated what we did before, now starting at a ridge stage. And if you culture those in vitro for 2 days, you get the extra layer of muscles and you get zigzags. So now you repeat it in the presence of the drugs. And the prediction would be, now the tube's going to expand longitudinally because you're no longer getting the second layer of muscles that prevent that expansion, and you'll fail to get zigzags. And indeed, you get an expansion longitudinally. So if you start with something that's 1 centimeter long, when you culture it, the control becomes 1.5 after 2 days, whereas these become 2 centimeters long. So you are constraining it if you have muscle, because this is blocking the muscle. And indeed, sorry. What's going on? I apologize. There we go. Thank you. And indeed, if you block the formation of the second smooth muscle layer, that is to say the longitudinal muscles, instead of getting zigzags, this is the zigzags in vivo, these are zigzags cultured, but if you culture with either of the drugs, then you don't form zigzags. So again, that second smooth, and the first layer of smooth muscle that's already differentiated is not affected by the drugs. It's only subsequent differentiation that's affected. All right, so we think that's what's happening with zigzags as well as the ridges. What about the formation of the villi? Well, the process of villus formation, sort of outlined here, is one where you get bulges forming along each edge of each zig and zag. So for example, here is a zigzag, and there are the bulges. So the bulges are not coming from the corners, they're coming from the edges. And these are the bulges that are going to give rise to the villi. You can sort of see that here in a series of different images. Here the bulges look like this, and they become larger, and larger still, and finally you end up with villi. So it's a continuous transformation from zigzags to villi. And as we said before, the timing of that is such that that occurs right when you form the inner layer of longitudinal muscle. So we do the exact same trick as before. We have zigzags and take it to bulge stage with this inner layer forming. What happens if you block the formation of that final layer of smooth muscle with either of these drugs? The prediction is these bulges will fail to form, and that's exactly what you see. So if you culture without the drug, you get in vitro the inner layer of longitudinal muscles forming, and you get bulges, whereas you are left with just zigzags if you culture in the presence of either of these two drugs. So we think we have an understanding about what's happening in terms of how you get villi morphologically in the developing chick. You start, and by extension, presumably, since they go through the same morphological steps, in the human and other mammals. You have the differentiation of smooth muscle in a series of sequential layers. you've got continued proliferation of the inner mesenchyme and the inner epithelium, and because there's a space constraint, you get buckling in a series of very constrained ways. I believe in conservation. I believe in everything Alan told us. I believe that animals, certainly as closely related as humans, cows, and mice, that all end up with villi should form villi the same way. Yet we're left with this description that says the mouse intestinal villi does not start with previllus ridges. Now my suspicion was because mouse embryogenesis is so much more rapid than human or cow, or chick for that matter, during these stages, that what's likely to be happening is you are getting the same processes, just that they're all compressed in time. So we wanted to just ask a couple quick questions about that. Say, number one, in the mouse, is the timing of the villus formation correlated with when the smooth muscle first differentiates? And is smooth muscle differentiation required to form villi in the mouse as it is in the chick or presumably human? And the answer is yes to both of those. So the timing when you get these villi forming in the lumen of, and you can see there aren't really zigzags, these just sort of get bubbles that then form villi, is exactly when the smooth muscle differentiates. And if you block the process with the drugs in vitro, in vivo villi culture, you get the same sort of villi without drugs, but with the drugs, you have a smooth lumen that expands and you don't get villi. So we think the same exact process is occurring with mice. And we're trying to investigate this more closely to try and understand if we can break that down into individual steps. But I now want to switch gears for the second half of my talk. So ultimately, you get these not bulges, but very long, thin villi. And more importantly, these villi are maintained not by mechanical tissue deformation, but by stem cells. And again, taken from the Nature Reviews genetics figure, there's this process where you go from all the cells being proliferating, self-renewing cells to a limited number of stem cells, first at the base of the villi and then in the crypts in the adult. So how does this occur? So the first question, before we say how does this occur, we want to think about when does this occur. At birth or hatching in the bird, the stem cells are all at the base of the villi. At the beginning, they're throughout the lumen. What happens to the stem cell niche over the course of forming first ridges, then zigzags, then bulges? And if you look, even as late as the late zigzag stage, now this is just a marker for Wnt signaling, which is an important signal in the gut for maintaining the stem cells. It's a niche signal. You see that Wnt signaling is present throughout these previllus zigzags. And you see that proliferation is continuous throughout the epithelium of the zigzags. As soon as you start getting bulges, the situation changes. When you start getting bulges, number one, the Wnt niche is now only at the bottom of each of these structures. Number two, proliferation disappears at the tips of the villi. And number three, you can look at a stem cell marker, LGR5, and show that it also is moving from the tips down to the base. The probe really doesn't work very well in the chick. So rather than a normal in situ, this is a single molecule fish where you're looking at the little dots. So the little dots down here are where LGR5 is expressed, and up here where there's an absence of dots at the tips of the villi is where it's not expressed. So the timing is right here between E14 and E15 in the chick when you lose this. Now, this loss of proliferation at the tip, how does that occur? We actually have a hint at that, and that comes from work in mice. So work from other laboratories in the mice has shown that there's a group of genes that's expressed at the tip of the growing villus that they call the Villus cluster. These cells up at the tip express PDGF receptor alpha, BMP2 and BMP4, and also a transcription factor, which is not shown on the slide, FOXF1. And that's previously been correlated with a downregulation, this is a BRDU staining, a downregulation in proliferation. So the interesting thing to us immediately looking at this data was we know something about BMP4 earlier in gut development. What we know is that BMP4 is initially expressed in the mesenchyme in response to hedgehog signaling. Both sonic and Indian hedgehog are expressed throughout the endoderm of the gut, of the midgut. And the receptor patched, which is in itself a target gene of sonic and represents where sonic and Indian are active, you can see there's hedgehog activity in a gradient going into through the mesoderm. And with a threshold, you can see that at fairly high levels of hedgehog signaling, you induce BMP4. So if BMP4 is downstream of sonic at an early stage, that suggested to us that maybe sonic is still controlling BMP4, in addition is controlling PDGF receptor alpha and FOXF1 from this and thereby turning on the villus cluster as a response to hedgehog signaling. So we look to see whether that might be the case. So first of all, this is now in the chick, our own data, sonic expressed throughout the endoderm of the gut. This is at the stage where you're starting to get villi or pre-villi bulges. Patched is expressed in response to hedgehog, especially at a high level at the tips. BMP4 is expressed especially at a high level at the tip, as is PDGF receptor alpha and FOXF1. So all of these genes that are downstream of sonic are expressed underneath cells expressing sonic. If you misexpress sonic, if you add sonic protein in vitro, you greatly expand the domains of their expression. So they are capable of being induced by sonic. And if you add an inhibitor of sonic hedgehog, cyclopamine, of course that doesn't have any effect on the expression of sonic itself, but it blocks sonic activity. In the absence of sonic activity, we knew that patched had to go away, but so does BMP4, so does PDGF receptor alpha, so does FOXF1. So it's looking good as if hedgehog is upstream of this villus cluster. But there's an obvious problem. The problem is sonic is expressed throughout the endoderm, yet these genes are only expressed at the tip. So why are these genes not coming on everywhere at an equivalent level underneath the epithelium? So we thought the answer to that might lie simply by thinking about the three-dimensional structures as they form. So initially, if you have a smooth lumen, you have the endoderm and a range of the secreted protein. So it's a gradient. Your protein's made in the endoderm. It diffuses. Various distances, you have different concentrations. But what happens if you start bending this? When you bend it into this is a cross-sectional shape of a ridge, of a zigzag, and of a bulge. This is the cross-sectional shape. And if you look at what you would expect to happen, if you started with something like this, what you would expect to happen is that these gradients would start to add up, so that you would get a concentration of signaling towards the tip, simply because the cells at the tip are now seeing hedgehog from three sides, whereas down here they're only seeing it from one side. And if you look at the expression of some of these genes over time, it really looks like that might be what's happening. Here is the PDGF receptor alpha, only turned on at extremely high levels of hedgehog that are only obtained directly under the epithelium at this early stage. As it folds and these folds start to come together, you start getting a bit of a higher expression where you're getting where you're getting these cells bathed from two sides. And when you get these pre-villus bulges, now they're seeing hedgehog from three sides, and that's where you get the villus cluster genes coming on. So I thought this model made great sense, and so I turned to my student and I said, Amy, what we got to do is, number one, we got to show that if you just physically open up this villus so that it doesn't fold up this way, but yet it differentiates the same length of time, that you shouldn't get this activation. And I think we have colleagues at MIT, we can microfabricate this little capillary, and if you use negative pressure, and I'm babbling away about how I have this vision I'm drawing at the blackboard, and she's shaking her head, she said, Cliff, give me a week, I think I can do that. This is why you have students. So students look right through some complicated MIT collaboration and come right to the point. So here's what Amy did. She went back to the bench and cut a donut out of an E15 gut. So at this stage, you're just starting to get these bulges. And she took this donut, which would look like this in real life, and that's a cross-section through these bulges, and simply turned it inside out. If you turn it inside out, what you do is you take these villi and you spread them apart. And now she cultured it for 24 hours and took a look by in situ hybridization. And this is what she saw. So these are the same age. They've been cultured the same length of time. But because this is now on the outside, where this is the inside of one of these circles, and because on the outside it gets stretched apart, you now, instead of getting a high concentration of the villus cluster genes coming on at the tip because of the high level of hedgehog signaling, now you're back to a situation that you would have normally encountered many days earlier. Okay, well, that's the villus cluster genes. What about stem cells, or at least proliferation? So she looked at proliferation in these donuts, and this is what you see. Normally, at this stage, by this point, proliferation and stem cell markers are only at the base of the villi. But if you turn them inside out, this is the same age tissue next to the same other adjacent tissues. The only difference is the geometry, and now proliferation is maintained throughout the epithelium all the way around. So I went back to Amy and I said, okay, that's great. We've shown that you can take a late stage and lose the formation of a villus cluster and lose the region of concentration of stem cells at the base. What about the reverse? Can we take a very early gut that is nowhere near the stage of forming villi and sort of suck it up into a microcapillary pipette? And now I'm starting to babble about collaborations again, and make villi prematurely just by bending the tissue and get this. And again, she sort of says, we don't need to microfabricate. Just let me handle it. So again, this is why you have students. So Amy went back to the bench. She took a gut at about this stage and flayed it open. So this is a ridge stage, and she cut it here and just spread it out. So this end used to be over here, so you see the ridges. This is it's about an E12 gut. And then she simply put a mesh screen on top of it and forced the cells, the tissue. Now they're constrained. They can't grow other than up into the mesh, and then form something that looks like an ear of corn. So this is 1 day later. This is E12 to E13. So you're talking about an E13 gut. This is way before you normally have villi. Villi are E15 or E16. So, and then she could say, what happens in that situation? What happens in that situation? These very premature protovilli. So these are ridges, and again, you have proliferation throughout the ridges. And now, when you make a pseudovillus, you get a region where you have a high density of signaling at the tip, and you start driving proliferation out of the tip prematurely. So we think we do understand both the morphogenesis of the villi and how that leads to localization of a proliferative population and of stem cells at the base. What we think happens is you start in the early gut with a series of differentiation events. Muscle differentiation occurs in waves in the chick and human. First a circumferential layer, then a longitudinal layer, then an interlongitudinal layer. And as this occurs, there's continued proliferation of the tissues that lie luminal to this. And because of those cells are proliferating in the face of this barrier, you get physical forces that deform the inner layers into ridges, into zigzags, into bulges that will form villi. Because that process, in addition to physically forming an outgrowth, are in addition changing the signaling pattern by bending a morphogenic field, you get local maxima of endodermally derived signals. Those local maxima induce genes that are induced only at very high threshold levels, such as FOXF1, PDGF receptor, and BMPs. And these, in turn, act to repress the proliferative potential at the tip, leaving proliferative potential at the bottom. In part, we suspect, although we don't have the data to show you yet, that the BMPs are counteracting Wnt signaling, which continues ubiquitously. So you lose the Wnt signaling at the tip, and you are only maintaining the niche further down. So this is really a story of morphogenesis, but we think that we can start to understand how morphogenesis in an embryo leads to setting up a system that will maintain morphogenesis in the adult, and that, of course, means stem cells. So I finally just want to thank again the people who did the work. Almost all of the work in my lab was done by Amy Shire, a graduate student. I didn't have time to show you mathematical modeling of the process, but we've extensively modeled this process with Maha's lab, Dr. Maha Devon, and his postdoc Thomas Talion at Harvard. And David Kaplan at Tufts University synthesized the silk tubes that gave us an artificial barrier in the absence of smooth muscle differentiation that also gave rise to similar sorts of effects. So I'll stop there, and I'll be happy to take questions.
Very nice talk. I do not understand how the zigzag pattern can be created by the first longitudinal muscle, because it's applied outside of the circumferential type of muscle and would not put any restraint on the growth of the inner layers. Right?
Maybe I should have been more clear. Muscle, because the muscle fibers form in a particular orientation, so if you had if you have skin is equally stretchy in all directions. It's isotropic. That's not true of muscle tissue. So muscle tissue, a circumferential muscle, because of the orientation of the fibers, prevents pushing outward, but doesn't prevent pushing this in this direction.
Right. But so that's the first, layer of muscle, right? So the second layer of muscle will be outside the first.
It is,
right?
but everything's knit together. So the inner layers can't pull up unless they can push up these circumferential muscles. And the circumferential muscles can't move up if they're constrained laterally.
So the first layer and second layer are physically connected in such a way.
They're all connected, yeah, very, very tightly. Yeah.
Okay, that's very interesting talk. And actually, I wonder whether the PDGF5 receptor alpha or BMP4 or the sonic hedgehog signal, they are essential for the microvilli formation by mechanical force according to your results?
I'm not sure I understood the question. So you're asking about the PGF receptor alpha.
Actually, yeah, those signals are important for making the microvilli formation. But in your mechanical systems, when you artificially make the microvilli
yes.
at the early stages, and so those genes are essential for making microvilli.
So if you make a microvillus at an early stage, I didn't show the data, but if you make the villus at the early stage, because of the high level of hedgehog signaling, you induce the PGF receptor at the tip. So you make anytime you make a villus shape with the epithelium and you get hedgehog signaling all around, you induce all of the villus cluster genes at the tip.
So my question is, if you knock down one of those signals,
so the roles of the individual molecules aren't clear. So other than hedgehog. So we are going to follow up and look at whether BMPs, for instance, are the reciprocal signal that interfere with Wnt signaling at the epithelium. PDGF, in addition to hedgehog, is made throughout the epithelium. So PDGF from the epithelium is undoubtedly signaling down, in addition to hedgehog, to the mesenchyme. And I actually thought that hedgehog might regulate the BMPs, but it would be PDGF or something else that was having a different role. I was actually surprised that cyclopamine got rid of all of those factors. But there are specific small molecule inhibitors of PDGF signaling. So we are planning on taking a look to see what the role of PDGF is in the process. We haven't looked.
Okay. And second question is, if you knock down the SMA, the smooth muscle actins, can you still see the microvilli formation?
We have not knocked down the smooth muscle actins per se. Which would be another way of not having it act as a barrier. But what we have done, just as an aside, is sometimes I get the question about muscle contraction. If you block muscle contraction, it doesn't affect the morphogenesis at all. It's the tensile strength, it's the physical properties of the tissue, it's not the contraction.
Okay, thanks.
So here,
yeah.
So in the physical constraint in which you have with the sonic hedgehog, right, you have these downstream genes target the tip. I'm just wondering if they are you're arguing that you have at the tip you have signaling from three sides, right? In the bottom you have one side. So do you think they're measuring the relative strength, from the tip to the bottom, or it's some sort of absolute value? So the specific, if you regulate the amount of sonic hedgehog signaling, do you see the tip expression expands or retracts according to?
So we haven't tried to play with it as a rheostat. All we've done is added a ton of hedgehog in vitro or blocked hedgehog signaling entirely. Increasing the amount of hedgehog in a more subtle way would be an interesting thing to do to try and look at the dynamics. I mean, other people have looked at hedgehog. One of the early role of hedgehog in the mesenchyme in a gradient is to specify different cell fates within the mesenchyme. So the different smooth muscle layers are actually induced by varying concentrations of hedgehog as you move. So one of the things that would certainly happen if you started changing the amount in the epithelium, the amount of hedgehog produced, if you doubled it, you would move out all of the smooth muscle layers. So that would certainly happen. But what it would do in terms of villus cluster might be worth looking at.
Well, I think your model can very nicely explain aspects of the timing and the general process of villus formation. But can it explain the actual high degree of uniformity of villi without some additional process to kind of make them very as uniform as they're observed?
And uniform in what sense?
Well, in terms of their diameters, their lengths.
So
Whereas this kind of mechanical, general buckling type model, it seems like there could be more variability.
So there's no variability at all in the width of the ridges, the height of the ridges, the width of the zigzags, the distance between a zig and a zag from row to row, or the early bulges.
But what
Then
makes
once
it so uniform is what I'm saying. What makes the ridge,
that comes the ridges being uniform is actually what you would expect if you have a uniform, is because the smooth muscle is differentiating uniformly around. So I didn't have time to go show the mathematical modeling that Maha does. But that's not just true here. That also happens, for example, in the trachea and other places where epithelial buckling. These ridges are always very uniform and very consistent. The subsequent uniformity of the long, thin villi, that's a different process. Once you form the bulges with the stem cells at the bottom, it's no longer physical forces. Getting from a bulge to a villus, now you're talking about effects that happen once you get a localized population of proliferative cells, and then I've got to turn to you for the uniformity. But in terms of the initial stages, the physical forces, that really is what you'd predict.
Hi, I'm Gang Li from Chinese University, Hong Kong. Really fascinating talk. I have maybe a comment, maybe a naive question, because you are using the intestine model to demonstrate the villi formation. But my question is, what about other part of the guts? I mean, do they have special patterning genes controls the specific part of the guts?
So there are two aspects to the question. One is, are there different patterning genes that make the small intestine different from the large intestine, for instance? Certainly, yes. CDX genes, Hox genes, and not my lab a long time ago did a little bit on Hox genes in the process, but regionalization of the gut is something that's been very well studied. Mm-hmm. Presumably, the different properties of the muscle, the differentiation, the rate of proliferation of the inner layers, and so on is different, because the villi that I'm talking about in the intestine are really a unique property of the intestine. You get something very different in the hind gut, and you don't get villi, of course, at all in the stomach. So the regionalization is obviously something very important that has to be layered on top of this to understand why the genes that control villus formation come on in the small intestine and come on either not at all or differently elsewhere.
Right. And the second question is about the muscle contraction force, as other speaker asked. During the development, I'm not quite sure about the chicken guts. Do they have any movement?
Yes,
in the embryo state?
They do, but as I've tried to explain a couple seconds ago in answer to another question, we can use blockers of contraction.
Yeah.
And so when you culture in vitro, you can take in vitro culture for 2 days a large segment of gut that doesn't have ridges and take it to having ridges, or one that has ridges but not zigzags and taking it to have zigzags in vitro, and that's totally unaffected by paralyzing the muscle. So the muscle contraction is not important in the process. They are contracting, but that doesn't seem to be an issue.
Right. Thank you. Thank you.
Okay. Oh, yeah.
Question. So the mesenchyme cell actually want to form villi. Mesenchymes also go into the villi. This mesenchyme cell actually provide a signal really could be controlled diameter, the uniform, the
So inside the innermost layer of longitudinal muscles, there is some mesenchyme that remains that does go inside and forms the core of the villi. There's almost certainly signaling back and forth between the epithelium and the mesenchyme at multiple layers and at multiple stages of development. We haven't seen any role for it in this process. That doesn't mean that there isn't one, it's not necessary to explain it. The physical forces can be, if you mathematically model them, are completely sufficient to give you these structures. And we know that if we get rid of these physical forces, you remove the process. But obviously there's epithelial mesenchymal signaling in this tissue as there is throughout the embryo, and it's undoubtedly very important for getting the right genes expressed in the right place.
You also shown, second question, you also shown that the Wnt signaling activity in the early progenitor cells. where the wind ligand come from? You know, that's a really big question. Also, you know, for the activated stem cell. Stem cell.
So embryonically, there are a number of Wnt ligands that are made, and so another aspect that we're going to follow up in the lab is trying to sort out which Wnts are really the critical ones here. But there are Wnts at this stage expressed by the epithelium, and there are Wnts expressed by the mesenchyme. Some are canonical, some are non-canonical. And several labs have sort of cataloged the changes in Wnt signaling. But they do seem to, the Wnt activity, at least as measured by the canonical Wnt activity, as assessed by CD44 expression, seems to track very much with where the stem cells are and where proliferation is.
Without further questions, let's thank Cliff again.
Thank you very much.
Very exciting lecture.